The plasma membrane is a selective permeable membrane, also known as a phospholipid bilayer, which is two layers of phospholipids. A phospholipid consists of a hydrophilic head and two hydrophobic fatty acid tails. The hydrophilic head attracts water, and the hydrophobic fatty acid tail repels water because water is polar. Only nonpolar molecules can diffuse through the plasma membrane without the help of a transport protein.
Molecules flow in and out of the cell if they are nonpolar. Polar molecules can if they have a transport protein to help them. We will go into detail later.
Molecules flow in and out of the cell until they reach equilibrium, which is where there is the same concentration of solutes inside the cell as there is outside the cell, and are flowing in both directions at the same rate. Molecules flow in and out according to their concentration gradient.
Diffusion is the movement of nonpolar molecules in and out of the plasma membrane. Then for polar and ion or charged molecules, facilitated diffusion is used, which means the diffusion through a transport protein. The plasma membrane is very flexible, and the lipid molecules will always assemble into a bilayer because the fatty acid tails are hydrophobic and will repel water while the hydrophilic heads attract it. To keep the fluidity constant, the number of saturated fatty acid tails can be increased or decreased, or cholesterol can be added or taken away. Within the plasma membrane, there are proteins that are used to transport or diffuse molecules in and out of the cell. Transport proteins are used for polar and charged molecules because the hydrophobic fatty acid tails repel them, so moving through the protein does not allow the fatty acid tails and molecules to come into contact with each other. Transport proteins are channel proteins, ion-gated channel proteins, and carrier proteins. Channel proteins have a channel through which water molecules or a specific solute can pass through. Ion-gated channel proteins are like channel proteins but contain a lid on the top that can only be opened when a molecule is attached to the lid. The lid will stay open, allowing other molecules to pass through as long as the molecule remains on the lid, but once it leaves, the lid closes. Carrier proteins ergo a change in shape that changes the solute binding site across the membrane. The molecule enters at the top of the protein and it is then pushed through the protein and out the other side.
Osmosis is the movement of water through the plasma membrane. Water is polar, so it cannot go through the plasma membrane. An aquaporin is the protein used to help water go in and out of the cell, since the head of the phospholipid in the bilayer is polar. Tonicity is losing or gaining of water by cell. There are three scenarios when osmosis occurs: an isotonic solution, a hypertonic solution, and a hypotonic solution. An isotonic solution is when the concentration of the water inside the cell is the same as the concentration of the water outside the cell. Animal cells prefer an isotonic solution because the concentration is at equilibrium. Plant cells dislike isotonic solutions because there is lack of turgor pressure, which means the plasma membrane does not have enough water to expand and put pressure on the cell wall, making the plant flaccid or wilt. A hypertonic solution is when the water concentration outside of the cell is lower than the water concentration inside the cell. This causes water to leave the cell and the cell will shrivel. In both plant and animal cells, this causes the cell to die. When the cell wall moves away from the plasma membrane, it's called plasmolysis. So neither cells prefer hypertonic solutions. A hypotonic solution is when there is a higher concentration of water outside the cell than the water concentration inside the cell. This causes water to go inside the cell, causing the cell to swell and lyse. Bursting only occurs with animal cells. Plant cells prefer a hypotonic solution. They prefer them because the swelling of the cell causes the plasma membrane to put pressure on the cell wall. This is what allows plants to stand up. In all of these, the fluid outside of the cell is the solute. Water molecules bond to all sides of the solute molecule to dissolve it. Once a water molecule is bonded to the solute, the water is no longer considered free water. Free water is what moves in and out of the cell during osmosis. Thank you for listening to this segment of our presentation.
Active transport requires ATP energy and goes against the concentration gradient because it goes from low concentration to high concentration. Active transport will never reach equilibrium. They also require a protein pump to function. An example of a protein pump is a sodium-potassium pump. In the first step, the sodium binds to the sodium-potassium pump. The affinity for sodium is high when the protein is at this shape. This step, sodium binding stimulates phosphorylation by ATP. In step 3, phosphorylation leads to a change in protein shape, reducing its affinity for sodium, which is released outside. The new shape has a high affinity for potassium, which binds on the extracellular side and triggers release to the phosphate group. In step 5, the loss of the phosphate group restores the protein's original shape, which has a lower affinity for potassium. Finally, in step 6, potassium is released, affinity for sodium is high again, and the cycle repeats. Another protein pump is the proton pump. The proton pump stores energy by generating voltage across membranes. It moves positive charge into the form of hydrogen ions. The voltage and hydrogen concentration gradient represents a dual energy source.
Bulk transport, also known as endocytosis, is when a single-celled organism engulfs large molecules or nutrients. This type of transport also requires energy. There are 4 ways this can occur: through phagocytosis, pinocytosis, receptor-mediated endocytosis, or exocytosis. Phagocytosis is when the single-celled organism uses energy to break its cytoskeleton so it can engulf a food particle. Then, with the organism's lysosomes, it breaks apart the food and absorbs its nutrient. Pinocytosis is when a single-celled organism engulfs extracellular fluid, for the molecules dissolve inside the extracellular fluid. Receptor-mediated endocytosis is the engulfing of a specific molecule using receptors to bond to it. The receptors are carbohydrate receptors attached to a protein at its base with a ligand at the receptor's end. The proteins with receptors on them cover the entire organism. The last type of bulk transport is exocytosis, which is the secretion of waste. Once the lysosomes break apart the food vacuole and the organism absorbs the nutrients, a secretory vacuole with the waste bonds with the plasma membrane and releases the waste.